Sir, what is base pair?
What is base pair? If you look over here, each one of these is a nucleotide, okay, over here. And when two nucleotides of, of opposite strands connect to each other through hydrogen bonds, we call this a base pair. So for example, A and T are, A from one strand is hydrogen bonded to the T on the other strand, and together A and T make up a base pair, because DNA pretty much is always in a double stranded form.
Okay, sir. Thank you.
Sir, why do we believe that protein synthesis doesn't directly come from DNA? Like, why is it not feasible, or why do we believe this?
So this is all a hypothesis, right? In 1958, these were the proposals, but these proposals were completely hypothetical. These were models. The same way DNA, as shown over here, is a model. Models are what you think is happening, but somebody has to prove it. It took 10 years to actually collect crystal structures of DNA to prove that Watson and Crick were actually right. And that was done by crystallographers. Fine. Similarly, there is something happening inside our cells. We can propose what is happening using the information we have gathered from literature by researchers who are actually doing the experimental work. But what is happening is what is happening. You cannot change that. Somehow evolution has reached a stage where certain biochemical reactions are happening and other reactions are not happening. So information is flowing from DNA to RNA to protein. Information in our cells is not flowing from DNA to protein. Now, maybe there is a universe out there in a planet where information is flowing from DNA to protein. But to the best of our knowledge, in the last 70 years of research, information is not flowing directly from DNA to protein, because there aren't any molecular machines which are inside the cell which can do this job. Okay. For example, if I ask you, can you make a molecular machine which can take a protein sequence, which is a series of amino acids, and make RNA? It turns out that there is no such machine inside the cell. Cell can make DNA from DNA, the DNA dependent DNA polymerase. Cell can make RNA from DNA, which is DNA dependent RNA polymerase. Cell can make RNA from RNA, RNA dependent RNA polymerase. So these machines, these enzymes are present inside the cell, and other enzymes are not present inside the cell. And why is it? It has something to do with fitness in evolution.
Okay, sir.
Any other question?
Sir, please can you repeat the X-ray and fiber diffraction method?
I'll actually go back to that in the next class in a little bit more detail. But what you should know very simply is that we are talking of molecules which are extremely small. You cannot look at them through a microscope. Their their size scale is to the tens of angstroms, which is pretty much what this uh, this movie is trying to show. Okay, they are very very small. And how do we how do we take pictures of these very small proteins and DNA? And the way we do it is we purify them so that there is only one species. For example, you can purify DNA, or you can purify this transcription factor, or you can purify RNA. So when you purify them in, let us say, milligram amounts, and you put them in certain salts and buffers, certain special salts and buffers, they tend to crystallize. And when they crystallize, the crystalline lattice is such, it is structured in such a way that if you point X-rays at a crystalline lattice, you get diffraction patterns. The X-rays bounce through the crystalline lattice and give you a sort of I'll show you pictures of what a X-ray diffraction image looks like. These images can then be interpreted to build the molecular structure of the protein or the DNA or the RNA you want to solve. So that is how crystallography is done. For fiber diffraction, you when you cannot form a crystal, the molecule refuses to form a crystal. It's not easy to do so. You can take fibers and you can point, which are basically wet fibers. These have a lot of water in them. You can point X-rays in a certain direction, which is basically along the axis of the fiber, and you can get diffraction patterns. And these diffraction patterns basically will allow you to interpret the possible spacing between and the possible length scales of the bonds inside the actual structure. So you can use X-ray crystallography as a tool to get diffraction patterns, and the diffraction patterns through a process which is complicated allows you to solve the molecular structure of the molecule which you are looking at, may it be DNA, RNA, or protein.
Thank you, sir.
Questions?
Sir, so could you please give a brief description of what a thermonucleate is?
So these are basically these are terminologies which are not used. Basically, it's a sodium salt of DNA.
Okay, sir.
Okay, so these are terminologies which modern biology does not use.
Okay, sir.
It's sodium salt of DNA.
I don't understand the meaning of double-stranded DNA running parallel to each other.
Okay, I'm doing a very simple thing over here, but it's for those of you who don't seem to be familiar with DNA. Basically, you have a strand of DNA. Okay, this is a very simplified way of making a DNA. And in order to make a double strand, you have a second strand of DNA. Okay, now we use terminology, and I'll explain this terminology, that a strand of DNA has polarity. It starts with a 5' phosphate and ends with a 3' of the phosphate backbone. And the second strand, which is forming this double helix, is anti-parallel. It runs in the opposite direction. Okay, so these two strands basically form the double helix with 5', 3', and 3', 5'. Now, what is this 3', 5' is something I'll explain to you.
So in one of them you wrote 3', 5', but in the second one was 3' and 5.
Yeah, so basically these are two strands which are anti-parallel. Okay, now let me try and explain them to you over here. Now, these are the 4 nucleotides of DNA. Okay, here is the nucleotide. So this is A, adenine, guanine, cytosine, thymine. Fine. Now, the orientation of this strand is 5' to 3', because, so now let me just start, see if you can understand this. So this is the nucleotide, this is the sugar, and this is the phosphate backbone. Okay, still remained yellow, that's fine. So now what is happening is, if you look carefully, you will realize that the backbone of a single strand of DNA is basically this phosphate backbone. Do you see this backbone over here? And the nomenclature is such that this is the 5' end of the phosphate, and the 3' end of this this nucleotide will attach to the 5' end of the next nucleotide, to the next nucleotide, to the next nucleotide, and so on and so forth. So you are connecting these 4 nucleotides A, G, C, T through the phosphodiester backbone. So this is the phosphate. This is the backbone which is connecting AGCT together. Now, what happens when you connect these nucleotides together using phosphodiester bonds? These 4 nucleotides are projected out from the strand. Okay, and opposite to these nucleotides, when you make a double strand, let's see if I can do that. So there will be a second double strand over here, and basically there will be a projection of T over here, C over here, G over here, and A over here. And this is in the orientation of 5' to 3', and this will be in the orientation 5' to 3'. And these two strands of DNA, the one on the left and the one on the right, will come opposite to each other, and they will form hydrogen bonds. And the hydrogen bonds, basically GC is a triple hydrogen bond and AT is a double hydrogen bond. This is something you will see carefully when you look at the structure of DNA. So these two strands are going to come together because of hydrogen bonds, which are forming between AT, GC, in for both these two anti-parallel strands of DNA, and which is what is shown over here. Here are the two strands of DNA over here, and they are coming together using these hydrogen bonds. So the dotted lines over here are the hydrogen bonds. Okay, and for each nucleotide, as I've told you earlier, this is the structure. There is a base, which is going to form the hydrogen bond in green. There is the sugar, which acts as a spacer between the phosphate and the nucleotide, and it's this purple part, the phosphate groups, which are going to bond with each other, forming the phosphodiester backbone. Is this clear enough? I can repeat this in the next class.
You said that the 3 prime and 5 prime have to do with something with polarity.
So it's directionality. Okay, the one strand is going 5. See, both strands are going 5 prime to 3 prime. So let me draw it again. Both strands are basically 5 prime to 3 prime. Okay, but they cannot make hydrogen bonds. They can't form a direct double helix unless one strand is 5 prime to 3 prime and the other strand flips and makes 3 prime to 5 prime, and it's only then that these two strands can come together to form a double helix. Is that clear?
Yes, sir. So the 5 and 3 are charges on the phosphate.
On the phosphate. It's nomenclature. It's the way it's the attachment of the phosphate to the sugar. And I think I need to define the nomenclature for you, which I will do in the next class. So, for example, here is the 5 prime. This is this phosphate group attached to the 5 prime of the sugar, and when it forms a bond, it will be the 3 prime of the sugar. I'll go through this nomenclature in the next class.
Thank you, sir. I understood.
So I have to end here. I will come back to DNA in the next class.